The aim of this experiment is to extract DNA from onion. Before we proceed with this experiment, we should know what a DNA is. The full form of DNA is deoxyribonucleic acid. It is a genetic material found in all organisms. The unit of DNA is known as nucleotide. Nucleotide join together to form a long chain called as polynucleotide. Within the cell, the polynucleotide are tightly coiled up to form a structure called as chromosome. The requirements for this experiment are chopped onion, 30 ml of water in a beaker, salt, conical flask, glass rod, dishwashing liquid, strainer, ethanol, spirit lamp, tripod stand and a blender. Chop the onion into tiny pieces. Take 30 ml of water in the beaker. Pour it into conical flask. Take 1 tablespoon of salt. Take 2 tablespoons of dishwashing liquid in the beaker. With the help of the glass rod, make a solution of it.
In this step, the salt and the detergent solution is added to the chopped onion kept in the bowl, and this in turn is added to a blender and it is blended for 2 to 3 minutes. Now the reason for blending is that for the DNA to be extracted or released from the onion, the cell wall, the plasma membrane and the nuclear material needs to be broken down. And this is facilitated by blending, and the salt and the detergent solution which we are adding actually helps to break down the cell wall and emulsify the lipids and the proteins present in the cell.
And the salt and the detergent solution is taken in the blender. It is blended for 2 minutes. The smooth puree is then transferred into the conical flask.
The next step, the mixture which is taken in the conical flask is then heated for about a minute with constant stirring. After a minute of heating, the conical flask is removed from the flame and it is then strained through a strainer. So the mixture is drained through a strainer. You can see a clear solution. So this essentially contains the chopped onion pieces as well as the salt and the dishwasher detergent which was blended into a puree along with 30 ml of water. Now some amount of ethanol is taken in another glass bowl and the filtrate is slowly added to the ethanol. So leave this for 30 seconds and you can observe the DNA being isolated as a thick white stringy mass. Now the reason for adding ethanol is that DNA is insoluble in ethanol and it can be isolated as a thick white stringy mass, as you can observe here. Thus we have isolated DNA from onion. Thank you.